Now let's take a look at signal transduction properties using the ligand insulin. The importance of insulin is that it is a small polypeptide hormone that is going to communicate information that is outside a cell to the inside of the cell. Now what we primarily mean by this is that insulin is going to be responding to glucose levels. Let's take a look at a graph in order to understand this. If we measure over time blood glucose levels, what we would see is that there is a standard amount of glucose in our bloodstream that is called the basal level. This is the level that glucose is maintained in the fasting state. So in other words, in between meals. Now when we consume a meal, our glucose levels rise, and then over time they return to this basal level. So this point here is mealtime, and that will enter us into what we call the fed state. Fed simply because our blood glucose levels increase. Now, if we take a sample of blood from the individual that has undergone this mealtime, what we would notice is that there is a small amount of blood insulin during the fasting state. Very small. But upon consuming a meal, the blood glucose level will rise, and there will be a concomitant rise of insulin. So this is an indication that the elevation, the increase in blood glucose concentration in the fed state is inducing a change in insulin concentration. This is causing an increase in the blood insulin concentration. So we have a hormone that is designed to communicate information based on glucose levels. So as blood glucose levels increase due to our meal, that is inducing an increase in insulin, the hormone insulin, which is going to communicate now to cells that glucose is high in the blood and available for taking up for generating energy. Now, how does this response work? The specific cells that are involved in detecting glucose levels are called beta cells. These beta cells are in the pancreas. Specifically, they are in small little islets of Langerhans. These are small little islets located in the large pancreas structure, and these constitute what is called the endocrine function of the pancreas. So the beta cells now are responsible for taking information on high blood glucose levels and transducing this information into high insulin levels. So how does the beta cell do this? Well, we need to look at the metabolism of glucose. So if this is a beta cell, we have on the surface of beta cells a specific glucose transporter, GLUT2. Recall that GLUT2 is a low affinity transporter. This is a facilitated transporter, but it's just not as high affinity as GLUT1 that we've seen before. However, after a meal, our glucose concentration is sufficiently high in the fed state that we have now surpassed the binding affinity for GLUT2, and the glucose is transported into the cell. The first reaction is the phosphorylation of glucose on the number 6 carbon, and this is done by an enzyme called glucokinase. This enzyme performs the same function as hexokinase that we've seen in red blood cells and in skeletal muscle, but it has a lower affinity for glucose that matches the lower affinity of GLUT2. Once we have our glucose 6-phosphate in the cell, this will be processed by glycolysis, the TCA cycle, oxidative phosphorylation, and the end product that we are generating is a large amount of ATP from this process, the oxidation of glucose. Now, if we look at the concentration of ATP, its concentration rises dramatically in the cell due to the oxidation of glucose. If we look at the ratio of ATP to ADP, we see this is a very high ratio. So the elevated concentrations of ATP are going to be affecting a change in the cell's physiology and behavior. Specifically, the ATP elevated concentration is going to be inhibiting an ion channel. This ion channel is responsible for transporting potassium out of the cell. Now, this is not the sodium-potassium ATPase that we've seen before. This is not primary or secondary active transport. This is just simply an ion transporter, facilitated diffusion, for transporting potassium. But this transporter is sensitive to the concentration of ATP in the cell. So with lots of glucose coming in, lots of ATP being produced, the channel is inhibited. Now, anytime we inhibit a channel that is transporting ions across the membrane, we have the possibility of altering the voltage potential across the membrane. And that's exactly what is going to happen here. There's going to be a change in the membrane voltage potential. This change in membrane potential is going to affect another transporter. This transporter is called a voltage-gated ion transporter. So the change in voltage potential across the membrane induces a conformational change in this facilitated transporter. In doing so, it will allow calcium ions to flow inside the cell. That will increase the concentration of calcium ions inside the cytosol. The effect of this is to cause the fusion of intracellular vesicles to the plasma membrane. Because this process of vesicular fusion with the plasma membrane, the fusing of these membranes, is calcium dependent. So as our calcium rises, concentration rises, the fusion of vesicles with the plasma membrane increases. And in doing so, now these vesicles will secrete their cargo into the bloodstream. And the cargo happens to be insulin. So the elevated glucose concentration in the blood induces changes in the cell, alterations of ATP due to the oxidation, increased oxidation of glucose. The increased concentration of ATP now inhibits the potassium channel, changing the membrane potential. Membrane potential is transduced information into the voltage-gated ion transporter. Now calcium flows in. Vesicles are now able to fuse with the plasma membrane, secreting insulin. Now when the glucose concentration in the blood, as we will see in a second how this happens, as the concentration of blood glucose decreases back down to the normal homeostatic levels here, the GLUT2 transporter, because it's low affinity, slows the activity and transport of glucose in the cell. By doing that, we reduce the oxidation of glucose, reducing the production of ATP. And by normal cell activity, the ATP begins to be used and decreasing its concentration. When the concentration of ATP is sufficiently low, the potassium channel is now allowed to activate. The inhibition is relieved. Potassium ions now flow. The membrane voltage potential is restored, and restoration of this voltage potential across the plasma membrane closes the calcium ion channel. Calcium concentrations decrease inside the cell, and vesicular fusion stops, thereby decreasing the amount of insulin being secreted by these cells. Now, once insulin is in the blood, the question is exactly what is insulin doing? Well, insulin's primary target that we are going to be talking about is skeletal muscle. The reason for this is because skeletal muscle needs an enormous amount of ATP. And it needs a large amount of ATP simply because of the contractile mechanism in skeletal muscle. Recall that the ATP is facilitating the actin-myosin apparatus that is inducing contraction. We'll take a look at this process in another video, but this apparatus requires a large amount of ATP. So skeletal muscle is definitely going to want to be able to take up glucose. Now the question is how does the cell tell if glucose is present on the outside? So we are assuming under these conditions, because we have high levels of glucose outside the cell because we're in a fed state, what that also means is that we have a very high concentration of insulin because the beta cells have responded to the high concentration of glucose in the blood. What this is going to do is signal to the skeletal muscle cell through insulin, our hormone, that there's a lot of glucose out there. So what we are going to do is transduce this information of insulin through a receptor. This is the insulin receptor. It is a heterodimeric protein. There are two alpha-beta subunits that come together and form a dimer. And of course, this is localized on the cell surface. When insulin levels are high, the insulin will bind to the dimeric receptor. Once insulin binds, what is activated in the receptor is what's called a receptor tyrosine kinase activity. What this simply is that there is a kinase activity in the beta subunit of each one of these dimers. That is going to induce the phosphorylation of the cytoplasmic tail of the receptor. This process is called autophosphorylation, simply because each beta subunit has a kinase and it phosphorylates the other. So this beta will phosphorylate this one, this beta will phosphorylate that one. Now the phosphorylation of this protein changes the look of the protein. And it changes the look of the protein such that another protein that is soluble in the cytoplasm is able to bind to it. So the phosphorylation of the beta subunits of the insulin receptor induces the binding of another component that is called IRS1, which stands for insulin receptor substrate 1. Once IRS1 binds to the insulin receptor, recall that the insulin is still bound to the receptor, so the kinase function on the receptor is still active. What that's going to do is phosphorylate IRS1. Now, once IRS1 is phosphorylated, this becomes an active second messenger. Now, this is free to dissociate from the receptor and transduce the signal from the receptor, which was induced by insulin binding, to other parts of the cell. And one of the main events of IRS1 signaling is to induce the fusion of a vesicle to the plasma membrane. This vesicle contains a glucose transporter. This glucose transporter, once it is bound, is translocated from these intracellular vesicles to the plasma membrane. Now it has the ability to take up glucose that is outside the cell. Now, GLUT4 is a very high affinity transporter. Being a very high affinity transporter, it can take up glucose very rapidly. And that's exactly what a skeletal muscle wants, because it needs a large amount of ATP. Once the glucose is transported inside the cell, it undergoes phosphorylation. This is done by hexokinase, which is a very active enzyme for glucose. First step in glycolysis. Two fates of this glucose. One is for ATP production to accommodate the large needs of the skeletal muscle cell. The other is for storage. And this storage is in the form of a molecule called glycogen. So by this activity, the skeletal muscle now can take the glucose up in a fed state, generate ATP as it needs, and also can store for later during a fasting state. Now, when glucose levels begin to decline after our fed state, entering our fasting state, the amount of insulin being secreted by the beta cells, of course, go down, decreases, and that means less insulin and less insulin binding to the receptor, and the receptor now becomes dephosphorylated by a class of enzymes called phosphatases. These phosphatases now inactivate both the receptor and IRS1. And this occurs in the fasting state, when insulin levels are decreased. Without this signal transduction, without an active secondary messenger, these vesicles are no longer fused with the plasma membrane. What actually happens is that these vesicles are These are retracted from the plasma membrane and return to vesicles. Thus, the GLUT4 in the fasting state is unable to take up glucose. So the skeletal muscle in the fasting state has to rely on the ATP it's just made in the fed state and whatever storage it has